Thank you very much for the nice introduction. For me, it's an honor to have the opportunity to share part of my PhD in a conference as EUCARPIA. The title of my presentation is Estimating Genetic Performance and Variability on FAR using ranking data. Before to talk about why ranking data, I would like to give an overview of the project that I am working with. The name is 1000 Farms. And the idea is engaging small-scale farmers in the plant breeding process. There is a special focus in collecting information on FAR trials. That information especially collected in the late stage plant breeding. And the idea is that the farmers themselves can collect the information and can compare the genotypes. I highlight in the map the regions that are focused. We can see that there are many countries of Africa. And then the question, why use ranking data for on-farm trials? Especially is because rankings are easy for small-scale farmers to evaluate genotypes. We don't need a special technology or special skill for them. They just need to give an order from what genotype they prefer until the less preferred. And especially if we are evaluating traits that are relevant for farmers, such as color, yield, marketability, even overall performance, for them probably it's easy just to generate ranking information. For that, we need a methodology that is called Tricot. I think was mentioned in a presentation yesterday too. In this methodology, we have an incomplete block design. It's a quite simple experiment where we have a set of genotypes that we want to compare. The farmer only received 3 genotypes and a small subset. As you can see here is a picture of a couple of farmers that are evaluating different genotypes. Why not 4? Why not 5? Because we want to keep easy for them. There is no any ambiguity, so they can generate information in an easy way. They can evaluate also different traits connected to different phases of the crop. The output will look like this one, where they will say which is the best and which is the worst. So it's only an order that we'll provide. So from one side, we have a methodology that can help farmers to generate data, ranking data. For the other side, we have some needs from the side of the breeders to make some decisions based on parameters or analysis that are required. And what I want to solve with my research is how can parameters required by breeders be estimated from that ranking data. So what I am proposing here is a framework to analyze this ranking data in plant breeding pipelines. And this framework, I will test using simulated data and real data. The model that I am using is called the Thorstonian model. The idea is quite simple. We have a set of genotypes, and there are a group of farmers that want to rank these genotypes. Those are the outputs that we can get from the Tricot trials. And the idea of this model is that those rankings are expression of an underlying continuous score that sometimes is called utilities. Those are in transparency because we don't see those values. We just assume that those values are there. And they are a function of mean utility and perception and environmental variation. And this mean utility corresponds with each item evaluated. And that in this case will be the genotype. So we can represent that a genotypic mean. And the relevant characteristics of this methodology is that we can use only ranking data to estimate those genotypic means. To estimate those parameters, we use the likelihood function. We only need the frequency of each ranking that we obtained times the probability of having that specific ranking. For make this likelihood function estimable, we need to add some restrictions. We have to set one of the genotypic means to 0. and also the residual standard deviation to 1. So that also will give the interpretation that, that we have from the parameters that we are estimating. From the ranking data, we can get the estimation of genotypic means, the variances associated to those estimations, and also the quasi-variances that sometimes facilitate the inferences that we can use on these genotypic means. So the framework that we are proposing is this one. We have the ranking data that farmers are generated. Then we use the Thursdonian model to convert this ranking data to continuous estimation of genotypic means. Specifically, we have those estimations, also the quasi-variances associated to those estimations. Then we can just run a conventional analysis using mixed model effects. In this case, it's relevant that we use the variation of each genotypic mean in the model. With this, we can estimate the genetic variance, and we can estimate the heritabilities. Even we can expand this model for run a multi-trade or multi-environmental model. So that's open all the possibilities of analysis once we do this conversion from ranking data to metric estimations. For the simulations, I generated yield as a quantitative trait with a set of genotypic means and adding FAR deviations and error deviation. The simulated data looks like that. From one column, I have yield associated to each FAR, and then I have the rank in orders. So here I am using the classical approach, using blues and blobs. That is my reference. And with the ranking data, I'm using the approach that I am proposing. I conducted different experiments, try to represent the condition on FAR, reviewing the literature, and also I try to make it stream to see how the methodology will perform. With the results, I was able to identify that we can obtain accurate estimations of genotypic means. But of course, we are losing information when we are using ranking data. So the variations associated to those estimations is higher. And also they have this shape that is like kind of a smile, where the string genotypes, the most preferred and the less preferred, has a higher variance. And that is something that is part of this kind of models, because those values tend to be in the same condition, in the same position in the sample. Then they are not too informative as those that I'm here. So they can be placed in different positions in the data. When I ran more experiments, I also noticed that when we have a high genetic variance and a low number of replicates, the algorithm, the framework doesn't work good to estimate genotypic means. And this happened because in the extreme case where a genotype always is placed in the same position, you can see that we don't have an optimal in the log likelihood function. So we can call this as a caveat of the methodology. Fortunately, in the cases of on-FAR trials, we are expecting that the genetic variance is not too much high. Otherwise, we need to increase the sample size to have accurate estimations. Regarding the estimation of genetic variance, we can see that they can be accurate if we can accurately estimate the genotypic means and the variances. In the case of heritability, is usually lower than the heritability of metric data. But this is expected because we have less information with ranking data. Also, we test this approach using real data. Those are experiments where farmers has rankings, and at the same time, they generate quantitative information of yield. In this 4 data set with 3 different crops, we see that the genetic correlation was high. But also we identified that in some cases the heritability was almost the same, or in some cases larger than in rankings compared with quantitative evaluations. That's opened the discussion that maybe small-scale farmers can identify some genotypic differences if they use ranking rather than they use quantitative traits. Of course, those are specific cases and depend on the ability of the farmers and the trait that we are evaluating. Some takeaway message is, with this framework, I proved that we can use ranking data to estimate genetic parameters under realistic on-farm conditions. The approach can accurately make estimations of those relevant parameters. And for future work, I would expect that we can replicate this analysis in multi-environment and multi-triad with multi-triad data. Even if we have information of estimations on farm and non-stations, also the idea is that we can calculate genetic correlations and do and see how the effects of experimental stations will be reflected in on-farm trials. Thank you very much. I'm open for questions.